Great, thank you very much. So, hello everyone. It's 3 p.m. So nice to meet you, and we are really thrilled to organize this webinar dedicated to the microbiota evaluation. It's a really trendy testing on every lip since few years, and we are really delighted to give you a presentation of the general information regarding what can be do for the evaluation of ingredients and cosmetics, whether it is in vitro or in vivo. So first of all, we will start with Mariana Caronca giving an overall presentation about the technological point of view of the microbiota evaluation. Then I will give you within 5 minutes the trends in the testing field worldwide. Then after we will go through the presentation of Stéphanie Badel-Berchoux from Byome Labs, then Sébastien Voisin from Phylogene, then Grethe Reichelt from Byome, and Albert Dashi from Sequential. At the end of these presentations, we will have the question and answers session. So don't hesitate to type your question in the chat. We will be able to read them and ask every one of the speaker to answer you directly. So I let the floor to Mariana.
Hello everyone. So before talking about methods, we have to set in place the regulatory scenery. So up to date, there are no specific regulations existing for microbiome-related skincare products. So brands must comply to the standard cosmetic regulations. However, in 2021, the International Corporation of Cosmetic Regulation published Microbiome and Cosmetic, setting the keywords that we use usually in microbiome. So setting a platform to understand what is going to be important in the microbiome-related skincare products. At the same time, different private organizations have started to create labels such as microbiome-friendly or microbiome respect. And these labels, while they're not regulatory, have no regulatory recognitions, they allowed consumers to have a marketing tool to communicate and give consumer confidence. So these labels usually are mainly in vitro based or ex vivo based, except for one label that actually only demonstrates the preservation of the skin microbiome in vivo. So mainly there are two main approaches, the in vitro and the in vivo approaches of testing. So they each have different advantages and limitations. So when we want to see the effect of an isolated bacteria, we're going to choose more in vitro because it allows us to have a more controlled and reproducible setting. And also it's faster and easier to make. However, if we want to see the real impact of the skin microbiota in the skin and have a more overview view of the microbiome, we have to see also the effect between the host and the microbiota. So this is, we're going to do more in vivo approaches. However, they have more interventional variability and are really time consuming. In the in vitro methods, usually we use culture-based methods. So as I mentioned, a big limitation is that only a fraction of the bacteria can be cultured. So these methods are quite fast and can give you certain answers. We usually use either reference strains or strains that we can obtain from in vivo sampling. However, if we want to see an effect and cultivate the need of cultivation, we can analyze also the microbial DNA directly obtained from samples. These methods could include qPCR, which is quite fast, or RNA sequencing, or next-generation sequencing, such as shotgun metagenomics. And for doing this in vitro test, first we have to be able to collect the samples. So in cosmetic studies, usually the samples are collected either through swabbing or through tape stripping. Swimming is quite simple. However, it only allows us to access the microbiota that is in the superior parts of the skin. Taste treatments is slightly more invasive and allows us to have samples from layers that are a little bit deeper in the skin. And there's a great tendency now to the use of at-home kits, which facilitates the participation in large scales. So actually, this is something that we can see a lot of brands utilizing. A great improvement in the skin microbiome testing was introduction of the next generation sequencing. These techniques have really profoundly transformed the study of the skin microbiota, enabling a much more detailed characterization of their diversity and composition, especially metagenomic shotguns that allows us to see the taxonomic and the functional profiling of the skin microbiota, and to understand who is there and what are doing these bacteria. Other techniques that are not culture-based are techniques of plate imaging. However, these techniques usually need staining, or need really to use the properties of, for example, polyphyllenes, which are produced by certain bacteria such as C. agnes, to be able to be detected with confocal microscopy. Also, a tendency today is towards the integration of multiple levels of biological data. So we use different techniques, so genomics, which we have already determined that we answer who is there, transcriptomics, which genes are expressed, proteomics, which proteins are produced, or metabolomics, where we can observe the metabolites that are produced by the bacteria. So the integration of these approaches are really offer a more holistic understanding of the skin microbiota ecosystem. Thank you very much.
Thank you very much, Mariana. So now I'm thrilled to give you more marketing and trends in the field of testing globally, what we think about microbiota now. So here we start. So this is the matrix I use to explain what are the sorry, si tu peux remettre à 5 minutes comme ça. Sorry. Yes, I use this matrix to explain all the parameters that are influencing the testing market. And then I review this global and systemic approach, and I was interested to see how we can see the microbiota. And I said, okay, finally, there is 4 points interesting. The microbiota evaluation is important for the digital experience and is really joining the digital experience in terms of application, nomad, because now there is trip people can do at home, consumer can do at home. Also the testing technology, because we use more and more omics techniques and EIA and database. So this is very important for the microbiota analysis. And I think the level of testing technology helps the microbiota analysis and these trends. The regulation constraint is also important because in France, the consideration of prebiotic, postbiotic, on every biotics fraction is different than in other countries, and specifically in China, at the moment, we don't know exactly what we have to complete for these claims. And the fourth element is the skin physiology, because now, after the gut, the microbiome of the gut, it's really the turn of the skin microbiota, the scalp microbiota. And I have looked also on the claims parameter that Skinobs is providing with through the world, looking at the importance of this claim in Europe, US, Southeast Asia, and for microbiome and microbiota respect, you can see that the leading place is US, and then Europe, and also in Asia, but South America and India is less interesting by this claim. I have checked in the testing platform what are the main. So it's not non-exhaustive list of service provider because there are a lot, but you can find among these one a lot of others, and you can see on the left you have the in vitro testing labs, and on the right the in vivo one, and in the middle the one that are providing both evaluations. And as the testing platform, we saw since 10 years the increase of the actors of this evaluation in this field, coming from the marketing to the R&D things. And so in violet, we highlight the companies that are offering really only microbiota analysis, and the bold one are present speakers of today. And then I was thinking it can be interesting also to see how the microbiota analysis is related. What are the related claims? And following the IFCC Congress in Cannes in September, and all the publications we can read about microbiota, it's amazing. The number is really amazing. We can see that the related claims to microbiota can be aging, can be sensitivity, can be also immune system, inflammation. We speak now about inflammaging, oxidation, the pollution also related to the skin barrier. Also, of course, obviously the acne-prone skin and the sebum, and as I said, the skin barrier, and also the well-being, because there are some recent publications that are highlighting the role of skin microbiota in the mental health. Here we can see how the microbiota analysis can be linked to the advanced evaluation typology. We can use also multiparametric measurement. It's also joined hyper-personalization. It's also linked to the in silico prediction, because one day when we will have enough data, maybe in silico prediction we will correct. And then inclusivity, because we can see in Chinese, Korean and Japan beauty the influence of microbiota, the gender between male and female, what's going on, the skin-gut connection also, prebiotic-probiotic fraction, local climatic condition, IA, data collection, and home device diagnostic, because we can use this patch to analyze at home. Here, it's just a screenshot of the testing platform. You can find here all the method for in vivo, and this is for assays. And here we just had I just had a look in the Cosmetic Textile News, this website that gather all the information about testing since 10 years. And when I searched for microbiota, I found 525 results. So that's very interesting, because last year we redesigned this website, and now you can use with the AI, your favorite AI tool. You can put the URL and ask for occurrence, for a summary, for whatever you like. So it's very powerful tool. And then, thank you very much for your attention. Next webinar will be 5th February about testing trends in India, and you will be able to see again this webinar on our YouTube channel. Thank you very much for your attention. And now I'm pleased to introduce Stéphanie Badel-Berchoux. Stephanie is graduated as a bioengineering engineer from Polytech and earned a PhD in microbiology and biochemistry. She then worked at Biofilm Control as laboratory director, primarily leading development projects related to antibiotic resistance. And in 2017, Stephanie structured the microbiology service activity while also participating in client relations and scientific communication. And these years, working with health and cosmetic industry players allowed her to specialize in the study of skin, oral and vaginal microbiota, and more generally in the relationship between microbiota, composition, and biofilms. As laboratory director at Byome Labs, she oversees the Clermont-Ferrand unit and is responsible for managing and executing client projects. Thank you, Stephanie. The floor is yours.
Thank you, Anne. Is the presentation visible for everyone?
Yes.
It's okay. Yes, thank you. So today, let's explore the relation between skin microbiota and the cosmetics and how to understand the microbiota, to measure it, and to innovate and taking into account the microbiota. So first of all, to understand the microbiota, I wanted to, with my initial idea, to put things into the context. It means that long after the influence of intrinsic factors like genetics and extrinsic factors like our diet, the exposome have also an influence, but a small influence on the microbiota compared to the first ones. And the cosmetics are some parts of this exposome. So, for example, we know that the microbiota is highly individualized and is specific of each person. And this is an example of a small population, very homogeneous, because it's our part of the Byome Lab teams. And we can see that there is a big predominance of C. acnes on our microbiome, but below to the dominance of the C. acnes, we can find our specialized microbiome print of each individual, and this is highly representative of one person. And to date, we know the influence of the prevalent strains. They are prevalent strains because it is also possible to cultivate them. So this is the four examples, like the Staphylococcus and S. epidermidis, that have a big influence in the skin microbiota because it participates to hydrate the skin by secreting some enzyme that helps to produce ceramides, but also participate to defend the skin by the production of antimicrobial peptides, and also to promote the proliferation, the keratinocyte, especially proliferation. We can find the C. acnes. C. acnes is very important to metabolize the sebum, and with this metabolization, it could maintain the pH, the acidic pH. With Corynebacterium, these bacteria are able to produce some fatty acids and small peptides to protect the skin. And finally, Malassezia that competes with pathogenic bacteria like S. aureus, but Malassezia also metabolizes the lipid from the sebum and helps to maintain an acidic pH on the skin. To date, we know that the preferred way of life of these microorganisms is the biofilm, because on the skin they can colonize the corneocytes, but also the sebaceous glands, and it allows them to persist on the skin even after mechanical action. But it also, the word is missing, it increases tolerance to external factors. And with the competition, the cooperation of the different microorganisms inside the biofilm on the skin, the sweat or sebum could be metabolized, and it also participates to educate the immune system by presenting very frequently, but in a small part, some antigens. And with this knowledge, because we understand the microbiome, how can we assess the different microorganisms that compose this microbiota? And the most important thing when we assess a product is to use the right model for the cosmetic product. When we want to study the microbiota, we have to verify 4 keys, because even if the microbiota is very individualized, we know that an healthy microbiota is very diverse. It means that a lot of microorganisms compose the microbiota. It's also a balanced microbiota. It means that the microorganisms are able to cooperate and to grow each other with a balanced ratio. But it's also important to verify that the biofilm form of these microorganisms are preserved, because if the biofilm is not preserved after a mechanical action, we can decrease the number of microorganisms on the skin. And a very important key is also the resilience. It means that even if a product has a big impact on the microbiota, if the biofilm is maintained, the microbiota has a very good resilience, and it's very important to keep a microbiota healthy. I wanted to show you an example where if the great model is not experimented for the product, we can incorrectly evaluate the product. For example, here is 2 models on the same product, and the objective was to prove the safety of a cleansing gel, so a rinse product, on the microbiota. First of all, if we want to verify the diversity, we can use a planktonic culture, and in presence of this gel, for 10 strains in the microbiota, we have kept a viability of only 1 strain, Staphylococcus capitis. And when we wanted to verify what is the impact of this gel on the co-culture of the 2 prevalent strains, S. epidermidis and C. acnes, we can find that even if the ratio is maintained between the 2 strains, the S. epidermidis population decrease for more than 3 log. It means that it's not a good result to maintain the balance. So we can conclude that this product should probably not be microbiome respect, but it's just because the product was not used as recommended for their application. So if we change the model and we work on the biofilm culture, we applied the product on an already formed biofilm as it's on the skin. And as it's a cleansing gel, we need to rinse it after a short time of application. And in this case, we have seen that the impact of this product with the appropriated application, so a short time of application and the rinsing step, we can see that there is no impact of the product, because when we numerate bacteria, each bacteria in a microbiota, there is a reproducibility of more or minus 1 log. And in this case, the product has no impact with its right application on the microbiota. So the viability is maintained and the biofilm form is maintained. So we can conclude that this product respects the microbiome. So how to innovate with the microbiome? To respect the microbiota, but sometimes to modulate it. There is the use of biotics, and I want to explain that the use of biotics should be considered only in case of dysbiosis, because there is no need to modulate the microbiota when we are with an healthy microbiota. But in case of dysbiosis, we can use probiotics as reseeders, and a lot of paper in literature, scientific literature, demonstrates their ability to reduce the inflammation and to strengthen the skin barrier. Most of the time, that's lactic acid bacteria. And it's also important to rebalance the population. Prebiotics should be also considered because oligosaccharides and sometimes polysaccharides should help to enhance the bacterial growth in the microbiota and to rebalance the population in case of dysbiosis. And here is some different skin affection where it's possible to act on the microbiota. For acne, we know that there is some pro-inflammatory phenotypes, and in this case, reducing these phenotypes should help, but it's necessary to preserve the diversity of the different C. acnes, because it's not all the C. acnes population that can be deleterious. In atopic dermatitis, it's important to rebalance the ratio of epidermidis by reducing the ratio of S. aureus in case of flare. The flare periods are the most problematic period. And in case of dandruff, it's due to overgrowth of Malassezia species, and we can verify the impact of ingredients or finished product to reduce this population, and always verifying that the diversity is maintained. And I want to extrapolate to the growing knowledge with the gut-skin axis. That is very important, but that's another subject. But in the next year, I think this axis will be also a good study axis in the research. And at the end, the innovation will be the hyper-personalization of the cosmetics. But to integrate the microbiota in this hyper-personalization dimension, it's important to have 3 key areas. And these 3 keys will be having some data. It's important to collect data to know the impact of the product and product ingredients or finished product. So collecting data, measure the microbiota of each person's, and then to put in relation the data and the microbiota of the person to recommend the best personalized product. And thank you for today.
Thank you very much, Stéphanie. Please stay with us until the end, because we will answer all the questions you can type in the chat. And now I am thrilled to welcome Sébastien Voisin, from Phylogene, the mass spectrometry lab manager. And a few words about Sébastien Voisin. Sébastien completed a multidisciplinary PhD in 2002 at Lyon University in France, and then joined a Canadian laboratory to learn proteomics techniques. After a decade in North America working for academic proteomics laboratories, he was recruited by a French CRO laboratory and eventually joined the CRO Phylogene in winter 2022. He has been working with a wide variety of biological models, including cancer biopsies, bacteria or bacteriophage samples, skin explants, 3D cell cultures, extracellular vesicles from different species, host cell proteins, and more recently on human microbiome metaproteomics projects. Thank you, Sébastien. The floor is yours.
Thanks. Sharing. Okay. You should see my slide.
Yes.
Welcome. So I'm going to talk to you about metaproteomics. So clicking here. So quickly, Phylogene as a CRO, we are providing services to both public and private laboratory, and we are notably proposing services in genomics and proteomics. But let's go about microbiota. So as you will know, we are all holobionts, all mammal, animal, and plant species are hosts for diverse microbiota. We are no exception. We are multiple microbiota at different anatomical sites. The more we learn about them, the more we understand where we are essential and involved in many biological processes. We are notably linked to numerous diseases, either as a protecting agent or when the microbiota is disabled or aging, it may actually lead to the onset of some diseases. But at Phylogene, we are proposing some solution to analyze the microbiota, classically targeted PCR or more widely metagenomic studies. We are also proposing metaproteomics analysis, where we are looking at the proteins in the samples to understand the microbiota. And generally, we are comparing two or more conditions, diseased versus healthy or treated versus placebo. All of these omics studies are generating a lot of data. So we are also proposing bioinformatics to extract meanings from all of this data. So why metaproteomics? This is how we are seeing the context. What metagenomics, we will get a microbial composition. We will say what could happen and which population is here. What metatranscriptomics, you will get the composition as well, if a little less accurately, but we also get the microbial functions, what seems to happen. What metaproteomics, we will say what make it happens, which are the proteins being expressed, not only from the microbial population, but also from the host, human or otherwise. And we will also get some data on the microbial composition, if less accurately than genomics analysis. And finally, among the omics, metabolomics will tell you what happens, but you will not get much data about microbial composition because metabolites are not always ordinary species specific, but you will not get much about functions either. To put it another way, in function of the service prestations being done, a targeted approach will provide data to make claims about the impact on the microbial population, but only for the targeted microorganism. A large-scale analysis like metagenomics will allow claim at the level of the bacterial or fungal population, and also claim about the impact of your treatment on the bacterial diversity, bacterial or fungal diversity. Metaproteomics will give you all of the above, more or less, and also give you some idea about the functions being improved or not by your treatment, so you can make claims about the effect of a treatment on the skin health, studying the skin microbiota. At Phylogene, we had the occasion to do metaproteomics on different forms of matrices, skin on the scalp microbiota using swabs, comedo, acne, if you prefer. Also some 3D skin model with the bacteria, feces. And we also did some study on the microbiota from plant, or a special case, the inoculum used in a biomass fermenting facility. To go more technical, generally we use this type of approach. We will go for multiplexing, labeling samples using a chemical label. This will allow us to treat a great number of samples. We did from a dozen to 100, maybe we can do more. Samples are fractionated to increase the number of proteins being identified, which led us to the next step, big data. We have plenty of data. It takes some time to analyze. A first step, generally, we use a full database, all known protein sequence from relevant bacterial and fungal species, and of course the host. When a second search, a second identification, when we confirm on a narrow database the identification, on calculate the relative abundance of the proteins between categories, treated versus placebo, etc. As an example, the results from a study we did on a treatment on skin microbiota between the start on 28 days after treatment. We found the indicated proteins from the host bacteria and fungi, and a number of proteins. We had the data to quantify them between the two times. When we did the bioinformatic analysis, we found that the treatment was not impacting the diversity of the community, microbial community, which is good. To continue on the bioinformatic analysis, our module, which we call Explore, is treating the data in this way. Starting from the metaproteomics data, we do a taxonomic analysis. We also try to do multi-omics. If we had data from a metagenomics approach, we will join it here. In parallel, we do a functional analysis where we are studying the pathway, the metabolic pathway being activated, modulated by the treatment. This analysis ends with a bibliographic search to analyze in the literature what is known about what we found. And we finish with a correlation analysis where the identified taxa and the signaled events are correlated with the function and the treatment. Or to put it another way, we start from the identified proteins. We go to the pathway being impacted thanks to a database about the relations between protein and biological functions. And we look at the literature to link this finding to what is known about these functions. And this will allow us to propose potential claims for the treatment. This is what we offer with a metaproteomics study. Thank you for listening.
Thank you, Sébastien, for your presentation. And we will have some questions for you and for others at the end of the presentation. Now I'm delighted to present, to introduce Grethe Reichelt from Byome. So I will let you the floor, as I don't have a specific mini biography from you, but because we have to change the speaker just from yesterday. So maybe you can I will be thrilled if you can just introduce yourself and give you some information about.
Yes, I can do that. Thank you.
Thank you very much.
Well, yeah, my name is Grethe, and I work as a Senior Business Development Manager at Byome. I worked there for close to 4 years now. Prior to that, I worked with Thermo Fisher Scientific for close to 12 years in different positions. And I have a PhD in molecular biology prior to that, as well as worked as a scientist in nutrition. So I've known Byome kind of since the very beginning. And today I'm going to talk about how we overcome the skin microbiome testing bottleneck and to introduce kind of a new standard for cosmetic innovation. So a little bit about Byome. We were founded in 2016. We are located in Oslo, Norway, in Oslo Science Park. And we are a microbiome testing service provider. So not a CRO, but we're a testing facility. So we work directly with other CROs, but also directly with clients. We started off with gut microbiome, but we have extended into skin microbiome and vaginal microbiome. And we have a proprietary method, which is called PMP, or Precision Microbiome Profiling. And this is a proprietary technology. But it's qPCR based, but I'll come back to that. So when it comes to skin microbiome analysis, what are the technical considerations? And the most important part here is really to find the right solution to what you want answers to or what you're looking into. First and foremost is resolution. How much resolution do you need? Is it genus level or is it species level? And that means, are you going to do 16S, shotgun metagenomics, or qPCR? And then, of course, it's which type of samples are you working with? Is it low biomass? And in general, for skin, most of it is low biomass, but there are differences depending on which part of the body that you're working with. And of course, as all of you probably know, there's a high impact of host DNA in skin microbiome samples. So there are some challenges related to, for example, if you have to do depletion. And this is the reason why prior many have chosen 16S, because that's really been the only affordable option in many cases. However, then you also have to do ITS if you're going to do the fungal part. And then it's quantification. Do you want relative abundance versus absolute quantification? And do you want to look at bacterial load? And bacterial load is super important for applications such as dandruff or atopic dermatitis and so on. So implication, cost, turnaround time, and accuracy of data. How important are these things to you? So to kind of introduce what PMP is, some of you may already know, but if we look at first taxonomic resolution for 16S, that would be, you can go down to genus level. If you do really well, you can do some species in addition. Shotgun sequencing, you can do species and subspecies, but that also really depends on how deep you go. So how much are you willing to pay to get those results? And I'll come back to the PMP. But when it comes to host DNA, 16S is not affected. Shotgun metagenomic is affected. But again, it gives higher costs because you have to sequence much deeper to get that result. And the problem in general, I think sequencing is great if you don't know what you're looking for. But the problem is, and it's been shown again and again, is that there are issues related to variability when you use different instrument, different chemistry, different database, even different version of the software. So unless you're really good at what you do and know how to deal with your data, there might be some challenges. And also, it's all relative quantification, both for 16S and shotgun, and the turnaround time can be quite long in many cases, depending on how networked you are or if you have your own facilities. Cost for 16S is low. Shotgun sequencing has been high. Luckily, all of this goes down. And that's why we introduced and went the path with PMP, because we can go, it's a targeted approach, qPCR, but we can look at a big panel of targets in one go. So for skin microbiome, we're looking at over 50 fungal and bacterial targets in one go. So we can look at all of them together. And we only use 5 microliters of your sample, so it allows you for a lot of other downstream applications if you should need. And we can look at proprietary strains, we can design, we can pretty much do anything that you want. And we're not affected then of host DNA or low microbial biomass. And I'll show you that from a published paper. And we get absolute quantification. So you get accurate, reproducible data, which means that you can actually compare between samples, between projects, and so on, which is something that you in theory can't do with sequencing, but that we still do. Turnaround time for us is hours. So that means that time from product to result, or development to result to launch date, all of that goes much faster. And that can save quite a lot of money for a company. And also the cost is low. And this is what I mentioned when it comes to the sensitivity. So this was done, and it was published by MIT, so Tammy Lieberman's lab in the US. And they compared shotgun metagenomic sequencing, 16S and PMP. And they were working with low biomass skin samples, so lower leg. So that's very dry area. And this has been calculated over to relative abundance to take into consideration that we compare with sequencing, and it's also genus level. But what we see here quite clearly is that for 16S, Cutibacterium kind of outperforms or dominates, so you're unable to see the low expressed species. However, if you look at shotgun metagenomic sequencing and PMP, they are both able to detect different species, even the low abundant species, and you get a more accurate picture of what's going on. So 16S has limited sensitivity, and in theory, it's not really recommended for low biomass skin samples. While PMP, as I mentioned, is on par with shotgun metagenomics, and it has the similar community composition and richness and evenness. And this excellent correlation is the same that we've seen both when we do the same with gut and we do the same with vaginal microbiome and so forth. And the reason for this is that the targets that we've chosen has been done with prominent key opinion leaders in the field, and every publication, everything. So we've done the work for you in a way. So we pre-selected the targets that's of interest. That's the main interest. And that's why we see that correlation. And when it comes to the applications that we have from our clients, it's quite diverse, and this is just a few, but this is more for the claims. So dandruff, we have a lot related to odor, and that also goes to some of our other panels. You have sun, UV products, inflammation conditions, you have acne, you have wound healing, you have atopic dermatitis, and you have diabetes type 2 diabetes, because that's also related to dry skin. And they can all use that one panel because it's so broad and it covers the whole body. And that allows you as a company to set up your system internally, and you can run the same panel over and over again. And instead of just choosing a few targets, you can see the full length of what's going on. And you can see, for example, are there some of these that are more proxy or drivers to the actual results? So it gives a very good overview of what goes on. And as I mentioned, we do custom analysis. We have subspecies, we can do strains, and we can also do digital PCR for those that need that. But everything in one single test. So which value do we bring then? So we are optimized for low biomass. Any type of extraction kit, we can get the swabs, we can get genomic DNA, it doesn't really matter. We have species and subspecies resolution. It's quantitative, so you get copies per microliter. So you can actually just just with your eyes, you can actually see the difference between the different samples or products that you're testing. And again, it's accurate and it's reproducible. You have fast turnaround times, so you get the results in days. And as I mentioned, data is clear and actionable. You don't need your bioinformatics pipeline. You can actually do it yourself. And it's very cost effective. So it's to accelerate your development and save cost. Who wouldn't want that, right? We work with a lot of CROs, so a lot of CROs actually offer our solutions through there because then they can do the sampling for you, and then you get the PMP in addition to other type of analysis. But we can deliver statistical reports. You can do multiple cohorts, time points. You have alpha diversity, richness, Shannon index, Simpson index, all of these, and you get the total bacterial load. And you also get the taxon abundance changes. And this can be implemented in your clinical study report, or you can create a separate report just for you. But it's an end-to-end microbiome service. So it's really tailored to your application and your specific needs. So, yeah, and new for 2026, we have an intimate care panel, which is kind of the intersection between skin microbiome and the vaginal microbiome, but it's also very odor related or odor focused because a lot of people are, for example, working with panty liners or products in that area. Then this is very, very relevant for that area. Yeah. And that's also over 52 species, and it's one test. So I think that's it for me. Anne?
Thank you very much, Anna Grethe. Your 10 minutes, exactly 10 minutes.
Yes.
Very accurate time. Thank you very much.
Thank you.
Remain with us until the end. And now I am delighted to introduce Albert. Albert, are you here? Yes.
Right. You hear me?
Nice to meet you. So now it's your time, and just few words about Albert. He's Chief Science Officer by Sequential, and Albert is the co-founder and CSO of Sequential. He completed his PhD in molecular genetics, epigenetics, and stem cell research at the National University of Singapore and the Genome Institute of Singapore in 2019. In 2014, he received the Singapore International Graduate Award from A-Star for his PhD research, and was also awarded the Young Investigator Award. He also won the Young Entrepreneur Shame Award by P&G for his innovative and business-driven ideas. Prior moving to Singapore for his doctoral program, Albert obtained his master in biomedical sciences at the University of Bern in Switzerland. Thank you, Albert, to be with us today. It's a real honor with all the awards you already got. So we are really we have the chance to have with us today. So I let you the floor.
Thank you.
I think your camera is off.
Is it off or should I off?
We can see your presentation, your PowerPoint, but we can't see you.
I can see myself. So I think
Peut-être
it's.
tu peux aller demander à Ilona ce qu'elle
You see the slide that I'm changing?
We can go like that.
Okay, my camera is on and I see myself and I see you as well. So I'm not sure.
Okay.
We just try to share. Do you see me now?
Yes, we can see you now.
Okay. I will share the full screen.
It was okay. My colleague told me it was okay.
Okay. Awesome. Again, thank you for the kind introduction.
That's not. This is only my question.
Thank you again for your introduction. I forgot some of those things. It's nice to be reminded sometimes. In terms of my background, I really appreciate this organization, and thank you again for the invitation. My name is Albert, and today I will be presenting Sequential and our non-invasive skin microbiome testing solution for really the ability to scale up studies and support you on more reliable data and claim substantiation. We know how a preserved skin barrier and protected health skin should be, and we know how a compromised skin barrier is with the specific conditions. And we do also know that the microbiome does play an important role in preserving the skin barrier. And then we know when is disrupted from intrinsic or extrinsic point of view and factors, then this will lead to a kind of a chain of reaction that will disrupt completely skin health. Skin microbiome has been with us for many years, and we have lived with them in a more symbiotic manner. But with years and evolution, things have changed. The way we live have changed, and we know how they regulate the immune system, strengthen the skin barrier, control the pH, and really are key in communicating with our skin on a day-to-day basis. But it has changed. Things have changed. What has happened? We live more in urban areas. Skin diseases that maybe 100, 200 years ago didn't really exist because we have changed completely the way we live. And this condition are also increasing. And new type of forms of this condition are actually evolving over the years. Consumers are searching more the microbiome, especially the post-pandemic. Things have really evolved quite exponentially. And at the same time, consumers are demanding for more science and evidence-backed products. And this, in parallel, has generated a spike of research that also has activated the grants and the institution and universities to do more research around the microbiome, the skin, and how does it influence the skin health. But for this, in terms of the claim, we know that more in human. I know there's a lot of word in vivo, which I like and appreciate that consumers are doing this more and understanding it. However, it's very important that we emphasize in the word in human, because most in a scientific community, when you say in vivo, sometimes you're also linking it to the animal work. But let's speak on the human data are becoming more and more important. And in order to substantiate really key products and what they do to your skin, we need to have relevant information on the skin on real time. So we know what is in vitro. However, as I said, we need more information on identifying the microbes on the real environment, which is the skin. And it's essential to do longitudinal studies. And we have worked with many of these studies, and subjects are very important due to the variability that exists across the individuals. But also very important to link the microbiome to the skin biology. So itself, what is the outcome from the host point of view? And link this with the proper statistics is very important. A lot of people misunderstand what a statistics is in the microbiome. Is completely different to what you're familiar with. And there is more rigorous statistics to be used. And really defining within the baseline what are responders and non-responders to really opt to the right claim substantiation. There are three main ingredients that we need for this to happen and really complete and offer a strong actionable insight and claim substantiation. One is a collection system. It has to be simple and scalable and non-invasive. Together with the integrated multi-omics formulation testing platform that can allow in more clinical study for actionable insight and really this claim substantiation. These three needs to really come together. And how we're doing it sequential. First, the collection system. We have developed a really unique way of collecting this low biomass that has been quite a key important point in this webinar as well, is how can we increase the amount of material we can collect from the skin. And we have tested many materials over the years and really developed a proprietary patch that allows us to, in a more faster, simplistic way, collect a significant more amount of molecular materials. And this, in case you can see here, we have DNA, which we collected for a specific processing. And we can see that we have a significantly higher collection system compared to the others. And most importantly, the variability is much lower. So this violin plot shows you that is more elongated, adding more variability. And we are able to compare with all the standard collection system. And this is very key for being able to use in different parts of the body. In addition, we are able to collect RNA, which is another molecule for analysis like metatranscriptomics we discussed. Most importantly is that you don't want the collection system to cause irritation itself that can confound the results. So we have also tested this with different methods, and we have compared with the standard collection system, which is the QDOM tape strip, and we can demonstrate that our system collects in a less inflamed and causes less redness, even when compared to the same amount, same surface area. So this is very important and is tagged along with the multi-omics platform. So at Sequential, we believe, yes, microbiome, but it has to come with the human component. These are very important aspects. And the way we do it is that we are asking the question that you want to answer and the claim you want to answer. And based on that questions, then we can target whether we go with the absolute quantification targeted approach or a more broader understanding of the bacteria's function. We are offering the metatranscriptomics, and I will give you a case study later. Fungal, the implication that it has, the fungi, especially on the scalp, all whole microbiome profiling. But all this together needs to be combined with the information that we can gather from the human. And with the human, we are able to also, with the same patch, and that's where the patch has allowed us to collect much more material when we can combine the different samples or the different detection. So in the same patch, we can use either one of the human component or microbiome component. Together with metabolomics, human gene expression, and protein profiling really closes the loop on what the human components and the type of analysis we can do together with the microbiome part. To give you an example here, what I've worked is, we worked in a study where we wanted to understand as well the differences between microbiome gene expression and the whole microbiome profiling. And this is just an introduction on the DNA. So one tells you, we already explained in this webinar, what is really present, and the other one, what is active, what is the microbiome is active, is expressing the gene. And it's very important to differentiate it because it will then link you to the claims that you can say. In one side, we really focus on this maintaining, preserve the microbiome profile, the amount, the abundances. We reduce the limits of growth. We promote microbiome stability and resilience. So all words that are key to what the detection is purely the presence of this microbe. On the other side, the RNA gives you really what modulates it, what changes, what are the pro-inflammatory modification. It does limit the pathogenic virulence of the microbe, promotes the functional balance of the microbe, really encourages microbiome recovery. So these are more stronger claims that you could push more if you were to use a technology like metatranscriptomics. And to give you an example of what this means, what are the differences is, this is just a collection between two different sites, completely different. One is the scalp, the other in the cheek. We have collected DNA and RNA from the same sites. And we, in collaboration with our the scientific advisors, we have profiled. On top, you see the metagenome. On top, you see the abundance of genes linked to the metatranscriptome. You can see that in the scalp, the majority of these are the presence. So this DNA is primarily Cutibacterium acnes, but the activity of the genes are mainly linked to Malassezia restricta. And this is very important because they are really more active. And same can be used for the skin. You can see on top, green being the Cutibacterium acnes, really present and highly abundant, like we always see it. However, when we look at the activity, you do see that this Malassezia restricta popping in some of the samples being active. So in some cases, the activity of C. acne represents the metagenomics of it. So the presence and activity kind of reflect each other, but not all the time. And why is this important? Because when you look at the same microorganism, but you put it on a two different environment, they are doing completely different things. So here an example of Malassezia restricta, and then we look at the gene activity when is either on the cheek and either on the scalp. And you can see that the genes that are really active here, color-coded, are involved in different mechanisms, so different gene activity. So on the scalp, it's more active in breaking down sugar and fatty acids, while in the cheeks are more focused on processing skin oils. This is really reflecting the unique lipid that is present in this area and the microorganism is feeding from. What we have done is we have extended this in using it in a more product testing. We looked at gene activity on a liquid foundation. And what we have done is we have done a longitudinal study here where we have tested 25 individuals. We can see there is makeup remover, and then we had a liquid foundation. What we have noticed is that with the liquid foundation, we see the S epidermidis here. I want to just emphasize on this week 1 and week 2. We see different groups at the baseline, so they're already different from the baseline. Some that are active in C. acnes, some of them are active in Phylogene restricta. And these are preserved. However, we see that after liquid foundation in one of the group, which is group 2, S. epidermidis activity has increased compared to the makeup removal. And how is this important? Because we wanted to look at the pathways that are involved. So we see a subset of pathways that are completely highly active, always present all the time, non-changing. But there are some pathways that are reflected and changing. And what you see here basically are color-coded in red, the more active ones, and then in light color are the less active ones. And you can see that there are some upregulated and downregulated. This is very important from a claim point of view because we can say more about it and we can push these claims more significantly, and it's very important to then define what the claims can do with this liquid foundation, whether it preserves the microbiome or really pushes the activity. Lastly, I want to just finalize with this is when we combine the different microbiome and the human part. So we have done bacteria profiling and protein and lipid profiling on the same location for these studies. Interestingly, we look at lip, and we tested a lip balm over a period of 2 weeks. And we did microbiome 16S, lipidomics, and proteomics. And the outcome is to generate this dry lip recovery pathway to understand, combining the three with biophysical measurements, the recovery of the lip health in this case. So we did microbiome 16S, so we see that there's no really changes on Shannon and Simpson diversity, but we do see a significant changes on metabolites compared to the dry versus non-dry after treatment. So in this case, we look at the dry lips only. We treated for 2 weeks with a lip balm, and we saw the effect that it had the lipidomics. It has also changed specific proteins that are linked to the barrier. But altogether, what we want to do in Sequential is really building these blocks of microbiome blocks, lipid blocks, and proteome blocks to link the molecular pathways and combine with the biophysical measurements, which are quite necessary to then understand the skin recovery pathway and be able to claim substantiation and new biomarker solution. We have done so by combining this, and in the claim substantiation for this project, what we did is we generated this recovery pathway score, and we see over transepidermal water loss and hydration, we see that there are some responders and some non-responders. There are some that low recovery, some high recovery. And we see that the high recovery had a lower transepidermal water loss, which is what you want, but it has a higher hydration. And we wanted to identify why these individuals are such and why the other one did not really work. To close everything, on Sequential, we really want to control this from end to end. So we're doing skin, scalp, vaginal, and oral microbiome, really from the beginning till the end. We also do the recruitment of the candidate. We have now over 50,000 different samples with the different metadata and biophysical. We have generated the Sequential discovery hubs really to bring new solution and actionable insight. And now we have formulations and parameters that we can combine based on the microbiome profiling and how they're linked to it. And each ingredient's connected and then pooled. We can understand it from a specific study how they're influenced. A simple, consistent molecular skin collection system paired with the multi-omics formulation testing platform and validated through clinical studies and deliver really actionable insight is very important. So this is the flow, this is the vision of Sequential, and it's a very important steps that we are now to really bridge the human with the microbiome. Thank you again for your attention.
Thank you very much, Albert, for this presentation. So now we have finished with all the 4 presentations. Very interesting, very different, and thank you, Anna, Grethe, Sébastien, Stéphanie, for your very specific presentation, and I can highlight that working with each CRO or each testing company expert in microbiome give you a special view and also evaluation of microbiota. And I think all the speakers has their own benefits regarding the test, and that's very interesting to see the panorama of what exists in terms of test and in vitro or in vivo. So maybe we can go through the question. Sébastien, Mr. Sébastien Voisin, if you can shut on your camera. Stéphanie, Badel also. So what questions? We had we had some IA agent, but there is a question for Stéphanie from Nikita, and he asked, 70% of cosmetics are thrown away. Where these figures come from?
Yes, that's a very surprising fact, indeed. It was revealed in 2018 by the independent IFOP Institute. And I was very surprised first. It's very, very big, but even if I'm a consumer of cosmetics, a moderate consumer, I tried to enumerate the number of cosmetics in my bathroom, and it was surprising. And I invite you to count at home, and this very surprising fact is real.
Not even for men, it could be surprisingly high.
Yeah, it's very, very high. Yeah.
One more question from Reichelt, Byome. What products specifically are consumers drawn to microbiome claims, please? Body washes, shampoo, moisturizer, cleansers, etc. Do you have this level of details versus appeal? So who wants to answer to these questions? What are the products what are the products you test mainly? Type of skincare or?
I can maybe speak up based on war studies. Most of the product on the skin side were cleanser and moisturizers that were tested in our side. When we speak in the microbiome, definitely we are quite clear on the body sides. So, we tend to be specified on the shampoo because what kind of shampoo, if you're reading a body shampoo or a, scalp shampoo, the questions behind that quite changes significantly. And the claims also change it. But primarily, shampoo, moisturizers and cleansers, or a combination of the two.
Yeah, I can follow up on that. We have the same, but a lot deodorants because it's a lot odor based, but also UV, so sunscreen, any type of sun product, but also creams for dry skin. So it's pretty much everything you can think of has kind of come through the lab, I would say.
Well, on our side, that I focus on functions, we get more things for treating skin disease or illness, like dermatitis, hair, scalp recently, so malassezia, things like this.
Okay, thank you very much. There is a question for Grethe, but maybe. So the question is: you may conclude on 50 species, but not skin microbiota with PMP, correct? How
Yeah,
can
well, we've
extrapolate effect on 50 space cells? Do you see the question, Anna?
Yeah, I see the question. I have them in
Okay.
front of me. Yeah.
Okay.
So
Okay.
there's two questions there that kind of overlap a bit. And yes, we can look at more targets, but what we've seen when we've gone through the literature, it's kind of the same players popping up. And you're thinking more of those that are actually actionable, something that you can do something with, something that you know has shown to have an actual function. So it would be valuable to get that information. So based and then on the feedback from the KOLs that we worked with, we found out that, okay, if we have these 52 fungal and microbial, then we have a pretty good panel that would cover most of the body. However, we also have specialized, so you can pull out just those target of interest if, for example, you're working with dandruff. But for a gut panel, we have over 110 targets. So it really depends. We have no limitations on that. And some do the panel and additional add-on with strains or mites, all these different things. So it really depends on which area that you're working with.
Okay. I see a question also regarding the pH balance. Is the pH balance can be supported for microbiome friendly somehow in claim perspective as many products in market? Is it correct as no specific regulation in place? Yes, I can just introduce an answer. Yes, here we can definitively study the microbiome quantitatively with this omics method, but there are also other instruments and multiparametric studies that are done since long time with the pH measurement, with the acne measurement evaluation with phosphorescence. So I will say that this measure of the microbiota and microbiome activity of the skin before and after the use of a product can be also complete by in vivo method using other more traditional testing. So that was the introduction I wanted to this answer, but maybe you have other comments.
Maybe I can just add something in respect to biophysical measurements as a whole. When we perform studies and we want to strengthen their claims and the correlation with the microbiome data, we always incentivize to do some level of biophysicals. I did not present it, but we recently looked at acne and skin and measurement of porphyrins and sebum production.
Yeah.
And we have a really nice correlation with our targeted subtypes of the different acnes, type 1, 2, and 3. And we see that the porphyrins abundance is significantly correlating with the type 1. And you can see that that link is there and is validated, suggesting that there is a clear communicational link even by looking at the abundances of this with the biophysical measurements. That gives quite strong strength claims. And if there is a product that reduces acne, you want to see that porphyrin levels changes accordingly. That's where the claims then becomes a bit stronger. If it is there, obviously, in the human context, it's not always as easy.
Thank you, Albert. I had another question related to the protocol design and the timing, because I've wrote that sometimes we can consider that the performance of skincare on skin microbiome can be quite quick. So in your experience, what would you recommend as the best time table for the protocol design?
As I said, Anna wants to speak maybe more.
No, I was just going to say it really depends on which product you're testing, right?
Yeah.
And what you're expecting is because that really differs depending on if it's cleansing or if it's a product for UV or if it's just, yeah. So that's where I think for all of us sitting here and presenting today, we like to have that discussion and interaction with the client beforehand so we can give better recommendations based on what you want to achieve. Because we've seen a little bit different things, and we also want you to succeed with what you do.
Yeah. Yeah, but I was thinking that, for example, for anti-wrinkle claims, we will say, okay, minimum D28 or D56 as minimum. But I was thinking, is there any difference considering skincare involved in probiotic with including probiotic and even for example atopic dermatitis. Shall we need to wait 14 days? Is it 1 day? Is it or is it long term?
Well, I can say on some studies that we did. So if you're referring of applying a probiotics, a live microbes on the skin, we haven't done many studies unless you are entering a clinical trial for that, because there's no cosmetic right now that can sell to you live microbes on a pot. But we have done studies, and the challenges is that the microbe that you want there to be and stay there doesn't always graft there or stays there for a long time. Because as we said, the microbiome, in general, the host microbiome is good at bouncing back. In this kind of study, we do so-called a dose response kind of approach. And then really we do the kind of, so to say, not pharmacodynamic, but we can use the same concept to understand how long does that particular probiotic stay there, and it really effectively modulating the microbiome around it. Because in a lot of probiotics studies that we did, prebiotics, for example, where you have just the pre, not the pro, what happens is that a lot of these formulations that are made are a fermentation process. And in this fermentation process, you have some, inside the pot, you have some DNA of the microbes that was used to actually make the fermentation from. And there is some residue of that DNA in the pot. And then when you do the sequencing, you see them, but it's not the host is actually coming from the product. So it is truly modulating, so you have to wait a little bit longer, and you can see that doesn't appear anymore. Normally standardized for product, we do an immediate, if it's a cleanser, we wanted to see whether there is a depletion of biomass immediately after the cleanser. And then we look at recovery. In our experience, the strongest cleanser that we used was a peroxide for hair treatment, for hair coloring.
Okay.
And then peroxide was like 6% peroxide. And then you can clearly see that there was a complete depletion. But interestingly, after 3 days, the microbe came back to the baseline level.
I would complete what you are saying, that it depends about the ingredients that are present in the product. For example, it's like for anti-infectious. Some ingredients will act in a short time, and you will see some inhibitory effect or no effect. And it's just like a one-shot activity. And for other ingredients, the impact will be with a long-term application. So some models will have their limits, and it will need long-term application with how to balance with the medium culture and the growth of the microorganism, not to support microorganism or to support the safe ingredients. It will depend. And we have to take in account the resilience of the microorganism that with their high tolerance will specifically secrete some enzyme and will communicate each other to tolerate more and more the stress that is generated by some ingredients, because not all the ingredients that is generated by the cosmetics application. So it's difficult to answer your question, and with only one model, one methodology, that's a big vision, and it depends.
Yeah.
I know it's not a good
Yeah.
answer,
It's
but we have
very-
no all the answers
Yes.
with the microbiome, this very
that's-
complex population.
Yeah, and that's why it's good to do either you know your product well, or if you don't, you should do a pilot just to test and see what you can expect, because that will help you to plan a better study afterwards.
Yeah. Well, I wanted to mention the study we did on Komedo, on acne. Our sponsor was telling us their treatment was so efficient. They almost have no Komedo left at the end of the treatment. So they were not able to give us samples. So, well, another factor, we have something at the end to analyze.
It's already 4:25. I think we will go really quickly in the remaining question. Once some are for Albert. Have you worked on skin conditions such as acne or atopic dermatitis? Yes or no? No answer. And
He's in mute.
Albert, you are mute.
I don't think you
Sorry,
heard.
sorry.
Yeah.
I wasted
Sorry.
I wasted precious minute. Yes, we do, and we are able to do it in different countries. As a follow-up to that question, I see she's asking.
Okay.
Singapore is pretty much in what is the Southeast Asian, but we can do recruitment in Japan and as well. And Europe in UK and New York in North America. The question is it possible to use the patch without the rest of the sequential pipeline? Well, yes, so the point of the patch is to be able to kind of plug and play with different CROs. And we can give the patch alone. There is some level of, protocols, but possible.
Okay. There is a question about do you recommend us any quick testing, just qualitative kit for microbiome testing, like lactobacillus survivor, in particular products like gel or cleansing products? Do you have some recommendations? Quick test like counting on in vitro plates. Maybe Mariana, you have a suggestion? No.
I can say that we've validated pretty much all the commercial available kits. Some are a little bit better than others, but in general, they work really fine. And I would always recommend if you're doing qPCR sequencing or whatever, do those that are stable in room temperature for 30 days because it's easier to ship and handle and all that. And they're really good. So yeah, you have a lot to choose from. Yeah.
Okay. Thank you very much. Thank you very much, everyone. Thank you, Albert. Thank you, Stéphanie. Thank you, Sébastien. Thank you, Anna, Grethe, for your presence today, for your great lectures. This webinar has been recorded and will be limitless in our YouTube channel, so everyone can see it again. We will share the PDF presentation of each presentation, and it will be shared in the article related to the webinar, and each participant will receive the PDF, the link to the YouTube channel, and also an exclusive white paper specifically dedicated to microbiome analysis. So thank you, everyone. Stay tuned, and hope to see you soon, the 5th February, about India. Thank you again, and have a nice day.